Hello everyone. In the last video, I discussed about pleiotropy, the first type of intergenic gene interactions, also called as non-allelic type of gene interaction, because in this type of interaction, generally alleles of different gene which is present on same chromosome or on different chromosome interact with each other that affect the inheritance pattern of characters. Now in this video, I am going to talk about next type of intergenic gene interaction, that is polygenic inheritance. It is also called as quantitative inheritance because in this type of inheritance, the quantity of the gene matter. Okay, actually pleiotropy and polygenic inheritance are completely opposite to one another. In case of pleiotropy, we discussed that single gene control number of different type of traits or character, but now in case of polygenic inheritance, many gene control single character. Okay, so remember that in pleiotropy one gene many character, while in case of polygenic inheritance many gene one character. Okay, so in this video we will going to discuss polygenic inheritance along with different examples. Okay, so this polygenic inheritance is also called as quantitative inheritance. Right? Because in this type, quantity of the gene matter. Many genes come together and then they control single trait or single character. Okay, that means many genes decide how a character will going to transfer from parent to progeny. Okay, we can define polygene as when two or more than two gene control single character it is called as polygenes or cumulative gene or additive gene. Okay, so you can define polygene as when two or more genes control single character or single trait, then they are called as polygenes or cumulative genes or additive genes, because there is addition of their effect. Okay, more is the gene, more is the effect. Okay, so if organism contain many dominant gene for a single character, then the character will going to express strongly. Okay, now in human we can find many examples of polygenic inheritance or quantitative inheritance, like height. Okay, then intelligence or skin color. So all these characters are generally controlled in human by polygenes or polygenic inheritance, like height of the human being or intelligence or skin color. Actually, in case of skin color, there are three different type of genes are present in human, and together they control the melanin production. That means ultimately the genes control skin color. Okay. Now, if in an organism, any character is controlled by two polygene like A and B. Suppose these are the two genes, capital A and capital B, and they are controlling X character. Okay, then in that case the F2 ratio or filial to ratio become 1:2, 4:2, 6:2, 4:1. So this is the second generation ratio in organism for the character which is controlled by two polygenes. Okay, and example is wheat, kernel color. Okay, so kernel color in wheat is controlled by two genes, and the second or the F2 generation ratio become 1:4, 4:6, 4:1 instead of 3:1 as that of the classical Mendelian ratio. Okay, so this ratio deviate from the Mendel's ratio, so we have keep this polygenic inheritance under the heading deviation, or it is come under post Mendelian genetics or neo Mendelian genetics. Okay, now if in organism single character is controlled by three genes or three polygenes like A, B and C, for example. Now in that case the F2 generation ratio become 1:2, 6:2, 15:2, 20:2, 15:2, 6:1. Okay, so this is the ratio of second generation if character is controlled by three genes. Okay, so remember that the 20 is in the middle. Now on the left side 1, 6, 15, and on the right side same thing 1, 6, 15. Okay, now this 1:2, 6:2, 15:2, 20:2, 15:2, 6:1 is the ratio for three polygenes, and example is human skin color or skin color in human. So in human being, skin color, as I told earlier, is controlled by three genes A, B and C. This three dominant gene is responsible for melanin pigment production, which ultimately decide skin color in human. Okay, now in this video I'm going to focus on two polygene example. Okay, that is wheat kernel color. Okay, so I'm going to discuss how exactly the kernel color is inherited from parent to progeny, and how I'm going to get, or we're going to get this ratio 1:4, 4:6, 4:1. Okay, so wheat kernel color is controlled by two genes A and B. Okay, because these two genes are responsible for red pigment production in the wheat, which ultimately control the wheat kernel color. If the wheat variety have both dominant gene, in that case the color will going to become red, and if a variety of wheat have both the recessive gene, small a for this gene, capital A is dominant and the small a is the recessive form of this gene, and small b is the recessive form of capital B. If the organism have this gene A and B, small a and B, in that case no red pigment production occur in the wheat and the color of the kernel become white. So we can find two different wheat kernel color, red and white, which is controlled by two polygenes A and B. There is a single character, that is wheat kernel color, which is controlled by two genes. So we consider this gene as a polygenes. Okay, this type of inheritance was discovered by H. Nelson in 1908. Okay, so he discovered wheat kernel color pattern, and in his experiment he crossed red kernel variety of the wheat with white kernel variety. Okay, so this is the parent generation and this is phenotype. Red kernel and white kernel is the phenotype of the parent generation, while the genotype was, for red we know that the genotype will be capital A, both the allele of same gene is dominant, and capital B, B also is in the dominant form. So in red kernel variety of the wheat, highest amount of red pigment produced, and the genotype is capital A and capital B. While in case of white kernel, no red pigment production occur, so that the genotype of this variety will be small a, small b. Now when Nelson crossed these two variety, red kernel variety with the white kernel variety, by using standard 3-step protocol of the Mendel. Now we know that this red kernel variety will going to produce gamete capital A, capital B, while white kernel variety will going to produce gamete small a, small b. So these are the gametes. Now they come together in F1 generation, in first generation, and the genotype of the F1 hybrid become capital A, capital B, small b. So this is the genotype of F1 generation, and due to this genotype, now this organism contains one capital or one dominant gene for A and one dominant gene for B. Okay, instead of all 4 dominant or all 5 small, they contain 50% dominant and 50% small. So the red pigment production become completely 50%, or the production reduced, so that the phenotype of this F1 hybrid become intermediate. That means F1 hybrid do not show complete red or complete white color. Instead, they show 50% red shade and 50% white color. Okay, because of that, the phenotype become intermediate. And then Nelson cross or self to F1 hybrid plant. Okay, now we know that this intermediate plant will going to produce 4 different types of gametes according to Mendel's law of segregation and independent assortment. Now this capital A form gamete with capital B, or capital A form gamete with small b. Similarly, small a form gamete with capital B and small a form gamete with small b. So these are the 4 different type of gamete which is produced by F1 hybrid plant. Now if we cross this plant with another same plant, then the plant will going to produce same gamete, that is capital A, capital B, capital A, small b, small a, capital B, and small a, small b. And if we make Punnett square, we can easily find all the possible genotype and all the possible phenotype. Okay, so I am making Punnett square for F2 generation. Okay, so now here we can make the final genotype capital A, capital A, capital B, capital B, then capital A and capital B, small b, capital A, small a, capital B, and capital A, small a, capital B, small b. Okay, so these are the first 4 genotype of F2 generation. Then we will going to get capital A, capital B, small b, capital A, small b, small capital A, small a, capital B, small b, and finally capital A, small b, small b. Similarly, in next column, we will going to get capital A, capital B, genotype, then capital A, capital B, small b, small a, capital B, and finally small a, capital B, small b. Okay, and in the last column, capital A, small a, capital B, small b. Okay, so capital A, capital B, small b, then capital A, small b, small b, small a, capital B, small b, and finally small a, small b, because both the gametes contain recessive genes. Okay, so now 16 different type of genotypes we will going to get in F2 generation. And now from this genotype, we can make the conclusion about the final ratio of F2 generation. Now this color pattern is mainly depend upon presence of capital or dominant gene in the organism. More the dominant gene, more will be the red color pigment and the color shift towards dark red. Okay, so this organism, that is with capital A and capital B genotype, will going to produce highest amount of red pigment and the color become dark red. Okay, now extreme genotype, that is small a small b, okay, will not going to produce red pigment. So in this case the kernel color will be white. So these are the two extreme genotypes. Now we will going to make the different combinations. Okay. Now here we get organism with all 4 dominant gene, 3 dominant gene, 2 dominant gene, 1 dominant gene, and finally 0 dominant gene. Okay. So we will going to count the numbers. Now the organism with 4, 3, 2, 1 and 0 dominant gene. Okay. So first, out of the 16, one organism have all 4 dominant gene. So 4 dominant gene will be present in one plant. Then 3 dominant gene, 1, 2, okay, 3 and 4. This 4 plant will going to have 3 dominant gene each. So 3 dominant genes are present in 4 plants. Then 2 dominant gene. Okay, so this organism is with 2 dominant gene. Same case for this. Capital A, capital B. Then this. So 1, 2, 3, 4, 5 and 6. Out of the 16, 6 organism will going to have 2 dominant gene, and finally these 4, number 1, 2, 3 and 4 will going to have 1 dominant gene, capital A, capital B and capital A. So 4 organism will going to have 1 dominant gene, and finally 1 organism will going to have 0 dominant gene. So the ratio become 1:4:6:4:1. This is the number of dominant gene and number of organism. Total will be 16. So we will going to get 16 different type of genotype. Okay, and now we are going to focus on phenotype of this organism. If the organism have all 4 dominant gene, in that case the color become dark red. Okay, so kernel color will be dark red in this organism. If the organism have 2 dominant gene, which is 6, in that case color become intermediate, like F1 hybrid. Now if the organism have 0 dominant gene or capital gene, in that case the color become white. Okay, so these 3, dark and white, is the extreme phenotype, while in the middle we will going to get 6 organism with 2 dominant gene. Now these 4 are with 2 dominant gene. Okay, sorry, these 4 are with 3 dominant genes, and now their color will be red. Okay, the color, kernel color in this 4 organism show intermediate. That means they are not completely dark red, or they also do not show intermediate color. The color is present somewhat in between these 2 phenotypes. Now if the organism have 1 dominant gene like this 4, in that case they will going to produce light red color. Okay, in this way we can make ratio 1:4:6:4:1, which is the standard ratio of F2 generation when the organism contain 2 polygenes. Remember that 1 means completely dark red color of kernel in wheat. Then these 4 organisms will going to have red color. Then 6 organism, maximum 6 organism will going to have intermediate color. Then the second last or the 4 organism will going to have light red and then white. Okay, if we go from top to bottom in this direction, color intensity goes on decreasing. Okay, at top we're going to get dark red, then intermediate, light red, and finally white. Okay, so if we give color to these squares, here will be the dark red kernel color wheat, and in this point or at this point we're going to get white color kernel, and in between this we're going to get sharp gradient of the color. Okay, complete red, darkest red, then red, intermediate, light red, and finally white. Okay, so this is the inheritance pattern of wheat kernel color, which is discovered by H. Nielsen in 1908, and the wheat generally show two polygene, capital A and capital B, which decide the kernel color. Okay, and actually both these genes have dominant as well as recessive form. Number of dominant gene directly proportional with the number of or the quantity of pigment produced. So these genes ultimately going to affect the quantity of the pigment produced, which finally decide the phenotype of the wheat. In my next video, I am going to discuss about the second example of polygene, that is when organism contains 3 polygene, and the example is human skin color. Thank you.